Hello, everyone, and welcome to today's webinar, Using Multiplex Immunofluorescence to Predict Clinical Outcomes in Solid Tumor Cancer. I am Marie Stone of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Akoya Biosciences. To learn more, visit akoyabio.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. This webinar is educational and thus offers free continuing education credits. Please click on the Continuing Education window at the bottom of your screen to obtain your credits. I'd like to now welcome our speaker, Alban BESSEDE, CEO of Explicyte. Alban, you may now begin your presentation.
So hi everybody, and thanks for the nice introduction. Also, I want to thank the organizer for giving me the opportunity to present the work that has been conducted in cooperation with the teams from Institut Bergonié in Bordeaux and Institut Gustave Roussy in Paris, as well as with the Centre de Recherche des Cordeliers and AstraZeneca. So the work I will present today has been recently published and is dealing about the evaluation of presence of mature tertiary lymphoid structure as a predictive biomarker for immune checkpoint blockade response. And this work has been achieved thanks to a new technology, so a multiplexed immunofluorescence approach. So here are my disclosures. I'm a shareholder of Explicyte, so a company working as a contract research organization in the field of immuno-oncology. So I will discuss today about cancer immunotherapy, which is a clear paradigm shift. So while most of the cytotoxic agents has been developed to target directly tumor cells, cancer immunotherapy aim at reinvigorating the immune systems to finally induce tumor cell death by the immune cells. In the 19th, the first evidence of cancer immunotherapy efficacy has been obtained thanks to the use of high dose of IL-2 for the treatment of metastatic melanoma, which demonstrated unprecedented response. But with some limitation, this treatment is effective only in a small fraction of patients and also is associated with significant toxicity. So at this time, cancer immunotherapy was not the path forward, and we had to wait several years of intensive research efforts until the discovery of immune checkpoint and immune checkpoint blocker and start the immuno-oncology revolution era. So 10 years ago, approximately, metastatic melanoma, the treatment of metastatic melanoma has been considerably changed thanks to the use of immune checkpoint inhibitors, so namely ipilimumab, but mostly with nivolumab and the combination of both, which derive really impressive clinical response, as you can see with tail on the curve, and with a strong improvement of median survival. So besides the use of cancer immunotherapy in the field of melanoma, and thanks to the success of this treatment, so immunotherapy has been tested in several other indications with difference in terms of rate of response according to the cancer indication. However, While this treatment approach is really effective, it still remains to be improved in terms of rate of response, as most of patients still die from cancer as they develop resistance and die. So immuno-oncology still have years to go. So the current challenge in the immuno-oncology field is the identification of predictive biomarkers that can allow a better selection of patients who might respond to immune checkpoint blockers, and also remain on the understanding of resistance mechanism, acquired or secondary, that can help to identify novel druggable targets. Okay, so I will focus the talk today on the evaluation of the determinant of response to immune checkpoint blockers, and the previous paradigm was mostly that T cells are the main cells involved in response to immune checkpoint inhibitor, but thanks to recent seminal work showing that B cells are also a key player in response to immunotherapy. So T cells are not the only key cells. B cells can be an important player. So this work, this several works that was published so last year in Nature, so demonstrated that B cell signature was associated with an improved response to immune checkpoint blockade in different cancer indications, so namely sarcoma, but also melanoma and renal cancer. So this evidence was obtained thanks to the transcriptomic analysis approach. And interestingly, in this paper, it was demonstrated that patients for which we found B cell signature was characterized by the presence of tertiary lymphoid structure, so-called TLS, which is a structure that favor the antigen presentation on T cells by B cells, and that can ultimately favor an anti-tumor immune response. So this tertiary lymphoid structure are organized with B cells and surrounded by T cells, but also with other key players like follicular dendritic cells. So the question we wanted to answer is TLS, the presence of TLS at the immunohistochemistry level, so at the structure level, is associated with response to immune checkpoint inhibitor in cancer patients. So as mentioned previously, so this work has been published recently in Nature Cancer, so thanks to a collaboration with several teams. And so the approach was possible thanks to the availability on a large cohort of cancer on sample from cancer patients, so a discovery cohort with 328 cancer samples from patients treated with immune checkpoint inhibitor, and it was a pan-cancer study with a different type of cancer, including, of course, lung cancer. So the first step of this study was to evaluate the presence of TLS thanks to a conventional immunohistochemistry approach, so combining the CD3 and CD20 markers, CD3 for T cells and CD20 for B cells, and we were able to detect in a third of patients the presence of TLS, so 105 patients with TLS and 223 patients without TLS. On patients for which we were able to detect TLS, we went more in depth to evaluate the maturity status of this TLS thanks to a multiplex approach combining different markers. So, for instance, CD4 for helper T cells, CD8 for cytotoxic T cells, CD20 for B cells, CD21, and CD23 for the evaluation of the maturity status. So after evaluation by expert pathologists, we were able to detect in half of these TLS positive patients, so 58 patients, the presence of mature TLS while in 47 the TLS were not considered as mature. And finally, we wanted to evaluate the association of the presence of mature TLS with a clinical outcome, so the objective response, but also the progression-free survival and overall survival. So before going into the implementation of this large panel on this large cohort, we wanted first to develop and validate this new panel. So this new panel here combining the different markers I listed and which are illustrated here as a multiplex but also as a monoplex stainings that illustrates that B cells, so CD20, are really surrounded by CD8 T cells, for instance, here, while CD4 are really rare, and that also you can see that you have some follicular dendritic cells and mature B cells as highlighted by the CD23 marker. So for the validation of this panel, we compare the density of this marker when they were captured thanks to the multiplex marker or through monoplex approach, and we were able to demonstrate that the density of the different cells were similar in both situations, and we then considered the panel as validated. So when looking at the association between mature TLS and the clinical outcome, we were able to demonstrate that patients with mature TLS had a higher rate of objective response, as highlighted here on the graph on the left part of the slide, with a significant difference. And interestingly, what is also important to depict is that patients with immature TLS, so still presence of TLS but in the immature state, has the same rate of response as patients without TLS. So meaning that it's really important to have a favorable environment within the TLS to elicit an effective anti-tumor immune response. So this association between the presence of mature TLS and clinical outcome is also evidenced here when you look at patients with a longer overall survival, which were characterized by the high rate of presence of mature TLS. These data were also reinforced thanks to the progression-free survival data and the overall survival data, demonstrating that patients with mature TLS have a better clinical outcome. So in the field of immuno-oncology, so the PD-L1 status is an important marker, and we wanted here in this work to evaluate if the presence of mature TLS was also associated with clinical outcome according to the PD-L1 status. So meaning in patients with PD-L1 positivity versus PD-L1 negativity. So for this purpose, we perform multiplex staining, so combining CD8 and PD-L1, so also thanks to an immunohistochemistry approach, and together with pathologists, there were a scoring of these samples, so with a TPS inferior to 1% and with TPS superior to 1%. And on patients for which the score was below 1%, we performed the same multiplex panel, and we performed the same scoring of mature TLS and compare again the clinical outcome to the presence of TLS. So here it's data, so from patients with TPS below 1%, and interestingly, what we observe is that independently of PD-L1, the presence of mature TLS was still associated with an objective response, with also the progression-free survival and with the overall survival. We performed the same approach, and looking at the predictive predictive value of mature TLS presence in patient with PD-L1 positivity, so TPS superior to 1%. And again, we compare we look at the association between the presence of mature TLS and the clinical outcome. And as you can see here, the difference is really impressive, as you can observe that patient with a mature TLS has a really better objective response, despite the limited size of the cohort here. So this is observable thanks to the objective response, but also thanks to the progression-free survival and overall survival. Even this is not significant, but again, due to the limited size of the cohort, but the trends are really clear. So altogether, this result demonstrates that the presence of mature TLS is associated with an improved clinical outcome in cancer patients treated with immunotherapy, and this independently of PD-L1 status, and so that must be considered for further clinical testing. So another marker that is associated and has been at least described to be associated with a clinical outcome on patients treated by immunotherapy is the level of CD8 infiltration within the tumor. And for this purpose, we perform an analysis. So here it's not CD3, CD20, but it's CD8 and PD-L1. But we perform the multiplex immunohistochemistry staining, and we perform then an image analysis that allow us to make tissue segmentation. So in green, which is not the tumor, the part which is not the tumor, and in red the tumor part, which were then segmented into cells and then phenotype into CD8 positive cells versus CD8 negative. On this basis, we were able to stratify patients according to the level of CD8 infiltration, and we were able to separate patients with CD8 high, so 163 patients, and patient with CD8 low, 165. On this, according to these two classes, we look at the impact of the presence of mature TLS on the predictivity. So here is the data of association with a clinical outcome, and in patients with high infiltration of CD8, we were able to observe again good predictivity of the presence of mature TLS, as illustrated here by an improved response rate in patients with mature TLS, and also as observed thanks to the PFS and OS survival curve. In the same way, we look at patients with a low infiltrate of CD8, and the results are really do not have the same trend, as there is not major significance in this fraction of patients. So it seems, in contrast to the PD-L1 status, that the presence of mature TLS is mostly predictive of the clinical outcome in patients with highly infiltrated CD8 tumor. But it's important to note also that patients with TLS are associated with a high level of CD8 infiltration that could explain also this difference in terms of predictive value of the TLS. So as a biomarker study, we wanted to evaluate the robustness of the approach using validation cohorts. So we had the chance to have access to two independent validation cohorts. So validation cohort A, with 131 cancer patients, same treatment, and also a pan-cancer study, and on which we use the same approach, so TLS scoring, TLS maturity, evaluation, and association with clinical outcome. The same design for the second validation cohort, so cohort B, with less cancer patients, 81, all treated with immune checkpoint inhibitor, and same strategy. And here I will present the clinical data associated with the presence of TLS. And here you can see that there is still predictive value of this marker as illustrated with objective response, the PFS and the OS. The same result was observed for the second validation cohort. So altogether, from this approach, this result demonstrates that the detection of TLS is a robust biomarker to predict response to immune checkpoint inhibitor, and had the advantage to be easy to implement, as it relies on available tumor samples. In parallel of this work, so the work that I just present has been based mostly on the scoring by pathologists, but we wanted also to look at the automation of the process thanks to the image analysis. So this work is currently ongoing, and so as you can see, so here is it's an illustration of two cases with detectable TLS. And so starting from this image, so after slide digitization, we were able to phenotype all the cells, and then thanks to a new algorithm, we were able to segment the tissue into tumor, but also into TLS. So this approach will be used to evaluate the association of image analysis based detection of TLS and the pathologist based detection of TLS, as to have an easy to implement tool to detect TLS in the clinical setting and routine. So from this study, some information I want to give you is that the presence, but it was quite evident that the presence of mature TLS is predictive of response to immune checkpoint inhibitor. So besides the use of the detection of TLS as a possible biomarker, it strengthens the fact that B cells play a critical role in response to immune checkpoint inhibitor. And the use of this biomarker can allow a proper selection of patients that might respond to cancer immunotherapy, and so that it's something that must be evaluated in a prospective clinical trial. And finally, besides pathologist assessment of TLS, so image analysis could be also an additional tool to simplify the process of TLS detection, and it's an approach which is under investigation. So finally, just I wanted to thank all the people who were involved in this project, and particularly Antoine Italiano from Institut Bergonié, who was a coordinator of this work with François Leloiret and all the Bergonié team. But this work was also performed together with Centre de Recherche des Cordeliers and Hervé Friedman, teams from Gustave Roussy and AstraZeneca, and also from Explicyte. So I will be happy to answer any questions from your side, and thank you very much for your attention.
Thank you, Alban, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. just click on the Ask a Question box located on the far left of your screen. We'll answer as many of your questions as we have time for. Let's get started. Our first question is TLS within the sample considered or only when within or close to the tumor?
Thank you for asking this question. No, it's important to consider only TLS when they are within the tumor lesion or at least close to the invasive margin. And so we defined a cutoff around 1 millimeter far from the invasive margin. So this is the first geographical aspect of the TLS positivity consideration, but there is another point that must be considered is also the number of cells within the TLS, so at least 50 cells that should be within this structure, so either B or T cells, but at least a minimal size of the TLS.
Okay, thank you. Next question. How were the images captured?
So yes, the image were captured thanks to the multispectral imaging platform from Akoya, so the Polaris system. And so first, of course, there is a technical advantage as this is a multispectral imaging platform. But another aspect that you can also capture from this presentation is that we performed staining on on a large number of samples, and this equipment is compatible with not high throughput but important throughput screening approach. So it's not. So it was really a big advantage to have this platform for this work.
Okay, great. Next question. Did you compare CD8 density obtained by image analysis with a score from a pathologist?
Yes. So thank you for the question. Yes, it's a point that we evaluated. Because for up to now the CD8 infiltration at least in clinical routine is mostly evaluated by pathologists with basic score, so highly infiltrated, moderately infiltrated. So there is a score, but with a limited, I would say, with a number of score, and so but still we compare this scoring with the density obtained by image analysis, and we obtain a good correlation. So meaning that the image analysis approach is quite robust.
Thank you. Next question. Do you think that the image analysis can be supportive for TLS detection and scoring?
Yes, we believe so. This work is currently under investigation, so we need to wait for the results. So as mentioned, so the important point is that we can consider a TLS only when within the tumor lesion or at least close. So ideally, it would be nice to have a tumor marker to help the TLS detection within this tumor area. But without it, it would be probably helpful to have validation by a pathologist. So again, it's an evaluation that we are currently doing. So more soon, hopefully.
Okay, thank you. Next question. In my current setting, I have a standard fluorescent scanner and I'm limited to 4 dyes. Did you use a specific cubes combination to image 7 dyes? Would it be possible to know which imaging platform did you use? Thank you.
Yeah. So yes, it's just what I mentioned before. Yes, we use this multispectral imaging platform, so the Vectra Polaris, which allows for 7 marker capture. So we cannot use a 4 marker. I mean, a system that can capture only 4 markers. So, or otherwise you need to decrease the number of markers you want to evaluate. Yes, and so this is also an important point to mention. So we use opal dye to reveal each marker, so which were compatible with the system we were using.
Okay, next question. What image analysis software did you use?
We use a system which is provided with the equipment, so it's Inform software that can allow very large type of image analysis. So we can, as mentioned, we can deconvolute the tissue in so at least the tumor sample within different regions, so tissue region, tumor versus non-tumor. And then we can use this cell segmentation for further phenotyping, either by machine learning. So this is based on artificial intelligence, but then also we can use. So this was not used in the scope of this project, but it's something that we are using for other projects. We can use an approach based only on the fluorescence intensity analysis, so similar to flow cytometry approach.
Okay, next question. Can you please explain a little as to how you validated your MIS panel?
Yes, so it's a two-step validation. So the first step, we validate each marker, and this validation is really based on discussion and exchange with pathologists, so expert pathologists that we have the chance to work with at the Institut Bergonier. And so once each marker are validated, they are implemented both in monoplex and in multiplex, and we evaluate the thanks to the image analysis approach, the density or the intensity of each signal in both situations. And as soon as we have the same pattern in both conditions, so monoplex versus multiplex, we consider that the multiplex is validated.
Okay, thank you. Next question. What criteria did you use to distinguish between CD8 high versus CD8 low? How did you decide on the criteria?
Yes, it's a really interesting question. So in this work, we decided to use a conventional approach. So we use a median value. So the patient with CD8 high tumor were above the median, and the other were below. It's an approach that we use in some condition, in some work, and for the work we can also make a different cutoff. For example, the 25% high patient. So I mean, patient can be only considered as high when superior to the 24% limit. But yes, it's really a subjective approach.
Okay, great, thank you. Next question. Aside from T-cell infiltration, do you have plans to examine the immune spatial neighborhoods surrounding the mTLS?
So no, we didn't look exactly at the tumor microenvironment around the TLS. So we have work currently ongoing on the evaluation on the structure of the exact structure of the TLS. So the composition of B cells, T cells, and other cells. For example, between responder and non-responder patient, but we didn't look at the correlation between the microenvironment around TLS, either they are mature or not mature. So yes, it's an interesting point that could be addressed.
Okay, great, thank you. Our next question has multiple questions embedded. So, hello, thanks a lot for your great talk. Could you please precise how you chose your threshold to perform survival analyses? mature TLS versus non-mature TLS? Also, was it easy to adapt your multiplex panel on several tissues from solid tumors? And last, how was designed your classifier to automatically detect TLS, criteria of staining only or also the area? And thank you very much.
Okay. Yes, good question. We didn't consider a cutoff for the mature TLS. We consider only the presence versus absence. If tumor sample had the presence of one mature TLS, it was in the class of mature TLS, and of course the opposite was without mature TLS. So it's not a matter of mature TLS density, for example, within the sample. So no cutoff or no threshold value. The other question is about the easy to implement approach for this multiplex, considering that it was a pan cancer study. So yes, it was. I would not say easy, but it was possible because we had pathologists working on this scoring. Because we didn't have, for example, a general pan cancer marker, so to define the tumor tissue versus non-tumor tissue. So this was possible because pathologists were able to score this different type of tumor. And last, how was designed your classifier to automatically detect TLS? So this point, it's quite complicated to answer because it's something that we are currently doing, and I guess it's based on the size of the TLS. So this is a machine learning approach. So it's not a black box, but we don't have all the algorithm detailed. So it's based on the size and probably also on the content of the cells. At least you have to detect organized B cells and with some T cells, either really organized around or within or mixed with the B cells. So this point is quite complicated to answer because we don't have all the elements.
Thank you.
Hope it helps. Thank you.
And next question. Your study focused on primarily B-cell biomarkers within the mature TLSs, but are you also considering interrogating other dendritic cell markers?
So yes, at least within the TLS, yes, we consider to look at additional marker for DC. But for instance, we use CD21, which is a marker for follicular dendritic cells, and but it didn't give better results when compared to the mature B cells feature. So it's something that we use, but we are using other technology approach.
Okay, next question. How thick should the slices of the tissue be, in your opinion, in order not to disrupt the whole TLS structure? And also, is there any correlation between TLS and tumor stroma formation or vascularization? Thank you.
So in the work that I presented, the thickness of the slide were similar for all samples. It was 3.5 micrometer, and in that case, I'm not sure it's the optimal thickness, but at least we were able on this condition to detect TLS. And then between TLS and tumor stroma formation, and yes, so this is a really a good point because we know that for example, high endothelial venules are really important for the formation of TLS, but in this study we didn't focus on this type of of structure, but it should be considered in the near future. But just one important point, I think this work is now can be considered as a really seminal work that demonstrates that TLS, so as detected thanks to the immunohistochemistry, because the previous work was really based on transcriptomic approach, so based on the signature. So this work paves the way of the importance of TLS in the response to cancer immunotherapy, for instance, immune checkpoint inhibitor. But many things needs to be done now to really understand how this TLS biology is important to control the anti-tumor response. And the point you raised, I think dendritic cells, but also this vasculature point is something that will be considered in the near future by our team, but also I'm sure by other people interested in this structure.
Great, thank you. Next question. Who made or stained the antibodies, Akoya or yourself?
Yes, this point. No, it's so we use the Akoya system to detect the primary antibody, but we use secondary antibody already conjugated with Opal. So I'm not the technical expert on this point, but I think it's based on this technology. I'm probably someone from Akoya can answer this point. Sorry, I'm not the expert for that.
Oh, that's fine. Next question. Can you describe the cancer types and stage for the patients in the training and validation cohorts?
So the cancer type, it's pan-cancer study, so solid tumor first, and carcinoma. So with a majority of lung cancer, of course, because it's where the immune checkpoint inhibitor are used in routine, and it was advanced disease for all cases.
Next question. What types of cancer have been investigated in your studies? Was the prevalence of TLS similar in different cancer subtypes?
So yes, it's again a pan-cancer study, and so different type of cancer. And yes, we observed a difference between the indication, but it was not a massive difference. The general trend was 30% of patients having a detectable TLS thanks to the immunohistochemistry and or immunohistorescence approach. But of course, we need to investigate more in depth this percentage according to the cancer indication using larger cohorts. Sorry.
No, that's fine. So it looks like we have time for one more question. Did you ever look at tumor microenvironment markers?
Yes, we look at different marker. It was not included in this work, and the analysis is currently ongoing. As in this work, we focus mostly on the marker within the TLS, not outside.
Okay, well, thank you again, Alban, for your time today and your important research. We would also like to thank LabRoots and our sponsor, Akoya Biosciences, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.